Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about gel electrophoresis common mistakes. And if you take a look at this gel, you would notice that we do not see bands here clearly, and some lanes even appear like is not present here at all. We do not see them. So what might cause this? Judging from the lack of other symptoms, it appears that the gel simply didn't stain well. Kumasi blue dye staining solution can become contaminated with SDS if it is recycled, and the dye becomes less effective and proteins don't show up as well as with the fresh dye. So what can be done in this case? As long as the proteins were precipitated in the gel by the acidified alcohol in the stain solution, the problem could be corrected simply by restaining with fresh dye. Sometimes we also can lose the whole lanes if we underload wells, so this can be another issue. So this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.